There it is. Okay, so bacterial pathogens today, bacterial symptoms. We're going to try to look at some signs. Sign is actual pathogen. Symptom is what we see, the outward reaction of a disease. So bacteria are single-celled organisms. They're extremely small. You're not going to see them with a compound scope, and you're definitely not going to see them today with the dyno light. So we're going to manipulate. We're going to look at large numbers of bacteria. We're going to see how they react. We're also going to look at some symptoms. All right, so let's start by talking about a couple of the very common symptoms that we see from bacteria. First of all, yellow halos. A halo is just a ring around the spot. Bacteria very commonly have yellow halos. Wait, let me see. I'm going to make myself a note here. Bacteria like very warm weather, so you're not going to see bacteria in early spring. You're going to see bacteria the later the season gets, so wet and hot. And typically you'll see bacteria on the new or not so old leaves. So remember, our fungal pathogens usually come from splash from the ground. They work their way up, so your older leaves are typically going to have the first symptoms when it's a fungal pathogen. Bacteria come the other way. So you'll usually see more bacterial pathogens from the upper side. Also, bacterial leaf spots will not have the tan center. Once the tissue dies, it just kind of collapses. So you're not going to see that defined spot for very long. Something called water soaking. We're going to look at water soaking today. Think of water soaking when there's just too much water within that tissue. So it breaks out of the cell. Like when you squeeze a leaf, what happens? So that would be water soaking. I have some samples today to show you. There are situations where we have a bacterial wilt. Bacterial wilt very common in cucurbits, so the bacterial wilt in melons and cucumbers. That's pretty specific on that one. There are some bacterial wilts in tomato and potato. And we have bacterial pathogens that cause cankers, particularly in peach. So not a lot of different bacterial pathogens across the board with the exception of vegetables. There are some bacterial spots, leaf spots in beans, in peaches, in a lot of ornamentals. So my examples are a little bit across the board. I'm going to try to go with fewer examples so we can get a little bit deeper. And this is one that's going to take practice. So bacteria are hard. So let's get started. So let's look first at halos. I, by the way, have looked at halos. I'm going to share my screen here. I've looked for halos. I had some really great examples at my house. Uh-oh. Guys, where is that? Here we go. All right, try again. It's over there. Click and drag. Right there? Yep. All right, I'm gonna try again here. I have two screens and I can only share what's on this main screen. I'm still not seeing it. Where did it go? It's somehow not. Sorry, guys. It's only giving me one PowerPoint. Let me see. What are you seeing? Are you seeing a screen on bacterial halos? I'm seeing the training website over here. I'm seeing that. That's crazy. It's not giving me the option of this one. Thank you for your patience, everybody. One day I will be tech savvy. And you share? Here we go. Now what are you seeing? Ha-ha! All right, I'm going to drag that back over. If you would all see my office, you would not believe just how messy it is. The custodian even stopped coming. Okay? How's that? Do you have this? I can
I see this trainings.
There it is. There it is. Okay, so I just want to really briefly talk about, this is what we call a yellow halo, and a halo just being a yellow circular lesion. So if you can you see my mouse? Yeah. Okay, so you see these yellow circular lesions. This is a deciduous magnolia. So this is a yellow halo. All right. Up here, this is a zinnia, this yellow halo. Very indicative of a bacterial pathogen. Not always bacterial. See the bean here? Not always a bacterial. The bacterial pathogens pretty clearly. If I see a yellow halo, I immediately start suspecting bacterium. And you'll see that there is not the white center or the tan center. That one looks like it a little bit, but you typically don't get the tan center like you would a fungal pathogen. It'll stay black throughout. So this is indicative of bacterial. There it is on tomato. And if you see the tomato here, you'll see there is necrosis here on this edge of this tomato leaf. We're good.
It's not showing the mouse.
It's not showing my mouse. Okay, I don't know. Aha, here we go. Are you showing my mouse now? No. Laser pointer. Here we go. Now do you see my laser pointer? Okay, learning curve here. All right, let's backtrack. This tomato leaf, this is a bacterial spot, but a lot of bacterial pathogens can go in through the edges of the leaf where the openings are in the edge of the leaf. So this can be indicative of a bacterial pathogen. This is similar to marginal leaf scorch that we see as a result of drought, but this one is a symptom that you would look for in particular in tomatoes. Here it is again. You see it in a marginal infection, but you also see the spots here with the yellow halo around them. Notice that none of these spots are really round. They're what we call angular. And again, indicative of bacterial infections. Angular meaning they're delimited by veins. So when they get to the vein, they kind of stop. So they can start out pretty circular, but when they start hitting those veins, it'll delineate or delimit that progression, and it kind of stops. So angular leaf spot, we're going to see that in a second with a sample that I have. Any questions here? So some marginal burn, usually think about how a water droplet would move. So bacteria are kind of oozy. They don't dry out and fly around like fungal spores. So they'll move with a water droplet. So once you have infection, which way will that water droplet carry bacterial cells? So sometimes you see them in the rib. Sometimes you see them on the edge. You'll usually see them clustered. If it's really rainy, they'll be more scattered. Or if you're using overhead irrigation, they may be more scattered. But often you can kind of see a pattern with those. All right. Any questions there? I'm going to stop sharing this screen. Any questions there? Okay. So that is the halo, and yellow halo, again, indicative of a bacterial pathogen. Let's look. I'm going to switch to DynoLite. Everybody good? I'm going to switch to my DynoLite. I'm going to have to, let's see how I'm going to share again. Okay, why is my DynoLite? I have to put it, so that's my main screen technically. All right, guys. Nope, not doing that. Here's my DynoLite. Yep. Okay, that's not what I'm seeing. I'm still seeing, okay. Oh, let's see. Okay. Okay, can I drag it over? Okay, Kim, can you swing that back around? So before this whole webinar series is over, I really am going to have to show you this office because we have a disaster here. Okay, so I've got my dyno light here with me, and I want to show you a couple of things. First, I want to show you an angular leaf spot. This is a hydrangea. It doesn't matter what it is. This webinar series is not about specific diagnostics. It's really focusing on the main concepts. So this is an angular leaf spot. And I am going to come down just a little bit more, and you can see the leaf veins. And you can see where the bacterial pathogen has pretty much stopped. Okay, sometimes, let me get my pointer. Sometimes you can see, this is hard. Okay, sometimes you can see kind of the raised edge. I'm going the wrong way. Sometimes you can see like the raised edge. There, notice there is no tan center. What?
We can't see what's under the dyno.
You can't see what's under the dyno light? Why? That makes no sense. Should I stop it and start it again?
Maybe stop sharing and go back into it.
Okay. We try again. We'll try one more time. Yeah. Okay. We're gonna get this one day. All right. So back again, here are the angular leaf spots, and they are delimited by veins. You see there's no yellow halo on this one. This is a hydrangea, and hydrangea are pretty typical for causing kind of that purplish reaction. It looks like a little bit like Cercospora, except Cercospora are pretty round. Bacteria are, again, delimited by veins. Let me see if I can show you again. I was struggling last time, and okay. Do you see the edges of this pathogen? See, they're a little bit raised. That can be common. You see the dark margin? It's a little bit raised if you saw it closer. Let me see if I can go down again. Is this the one I want? Which one do I want? See that raised edge? There we go. Kind of see that raised edge and see where the spot stops at that vein. There's a little bit of water soaking that's coming forward. I'll show you a better water soaking so you can understand it. So this is bacterial. There is no tan center. It is just kind of dark, and once the tissue collapses, it's gone far. All right, I'm going to come back up and show you that whole leaf again, because you obviously missed it the first time. Oh, there we go. So this is a bacterial leaf spot. This one is on oak leaf hydrangea. And remember, we were talking about the edges of leaves. Here you go. Look at the edges of this leaf here. See the edge? See where, think about water droplets moving through. Look where the leaf is creased. So that would be pretty typical. All right. So what I think I want to show you next is to look at the bacteria themselves. So we're going to look at what we call streaming. And I am going to make this attempt on camera. I need my slides. I had them wrapped up in a paper towel. Okay, hang tight, everybody. Yeah, there they are. And I have a bottle of water. All right, so we're going to look at what we call streaming. Bacterial pathogens will ooze through. We're always aiming for the margin of a spot. I'm going to come down a little bit. We're always aiming for the margin of a spot. So your bacteria are most prominent in the edge, in the marginal edge. Where is my... So in the marginal edge, like here, okay? So we are going to cut a very small piece. Our aim is to put it under a microscope slide. We're doing this with a dyno light, so you're not going to get the perfect scope view. So that's what I'm aiming for. This is something that you can do in your office. So I'm going to make this cut small as possible. Sharp blade is very important. Small as you can. I usually want more than one. Where are my slides? So I'm going to show you what I'm doing here. I'm going to try this. So there's a microscope slide right on the side of you. This is a microscope slide. All right, I have a bottle of water, and I'm going to make a drop with my finger, and I'm going to put a drop of water on the slide, or 2 drops of water, because what I am aiming for is for this to stick. A pair of tweezers would be extremely helpful. Okay, I've got butterfingers today. And so this sample would come right down. Right down there, okay? So I'm going to add a little bit more water there. All right, so you would do several of these, and I'm going to do that probably a little bit more off camera just to make it less awkward. I'm sure our trained pathologists are cringing right now because this is very, very crude. So microscope slide, and then I have some cover slips. And we can give you any of these if you want just a few. So I will put the microscope cover slip. Let's see if I can come up for you. And I'll put the cover slip right there. You see the water? You've got to have water there. All right. So our aim is for the streaming. So now I'm going to plug in. I have a light source. We have tried this with the light source being the back of a phone, a camera. So let's do this. There's my light source. Let's see if we can get this to do, Kim. Joy of awkwardness, right? How's that looking? So what we're going to do, we're going to be looking now for cloudiness. Let's see if I can get it out of this. So we're looking for cloudiness coming out. And if you look, this is going to be more of a time-lapse type project. You will see, in particular, let's see, right here. See that cloudiness coming out right there? All of this cloudiness, that's bacteria oozing out right there. All right, so kind of keep your eye out on some of the other sections. Where is it getting cloudy? Where is it changing? That one on the left is a really good one. Can you see it from your camera, from your screens? So that's a good one. I expected something from here, but my blade wasn't sharp enough. That's another thing. You need a very sharp blade, because if you've got ratty tissue, it's hard to differentiate where you've got kind of that move tissue as opposed to the actual streaming. We've got streaming coming in right here. No, a little bit further around. We've got streaming coming in right there. You see it getting cloudy. And you want to look at it pretty quickly because then it'll stop. There's a bubble right there. Oh, right there. That's a bubble. And that bubble is probably affecting. I expect that there would be really nice streaming right there. So these are clusters of cells. The bacterial cells are coming out of this plant tissue, and they're accumulating. All right, this is the this concept is once you adjust your eye to it, it's pretty easy to see. Okay, but you have to get your eye used to it. Some practice. You might want to have something that, say, the diagnostic lab has confirmed to be bacterial, and then get just maybe a fungal leaf spot and look at that and see that difference. The small piece, the one to your left, you'll see you've got some streaming coming around the top and then around that side. So it's kind of starting to accumulate. You really can't see here because of the ratty tissue. I would expect that there would be some right here. So a good blade. And of course, we have some coming in right there. Can everybody see that? I can't see the chat box right now because I'm sharing my screen. But if you want me to do another piece, let me know, so we can easily do some other pieces. So what I do want to show you, I'm going to lift this up and I want to show you really quickly. Remember we discussed, let's see, we discussed, I'm taking my slide off, this light fixture that we had bought. Can you see it there? It's just a little plug-in light fixture. This comes from the Dino Light Company. Let me turn the light off. This comes from the Dino Light Company. That's not working, is it? Where is that? There we go. It's just a little light block. And what we did yesterday is we flipped a cell phone over, turned on the flashlight to the cell phone, and we set the slide right on top of that. So just a backlighting of some sort is all you need.
We have a request to do another piece.
Request to do another piece. Let's see if I can do that again. So I'm going to start. Here we go. There we go. I'm going to use a better blade. So I'm going to use just an old-fashioned razor blade. All right. So I'm going to move it just a little bit so I can handle it. Small pieces. I'm going to get my slide back again. And I'm reusing slides, which is a no-no in the lab, but I guess it's going to work today. Let's see. Here's my slide. And I'm sticking my finger in a water bottle, and I'm putting water there, wetting my blade, because that will help pick up this piece of tissue. It's supposed to anyway. It's not happening today. Please use tweezers. I did not go down to the lab to get tweezers, which I really should do. You see that piece of tissue there? I'm going to cut one more. Then I'm going back to this point and I'm wetting it. I'm picking up this little tiny piece of plant tissue, attempting to. There it is. I've got two pieces. I'm going to put one more. Don't put too many because then your slide, your cover slip won't fit on it. Okay. You should easily be able to get microscope slides. If you cannot, again, I'm happy to share. And no more than you're going to do this. You can just reuse. All right. Where did I put my cover slip? So my cover slip is going on. All right. I'm replugging my little light block for you. And I'm going right there under the Dino Light. I'm going to lower my Dino Light. There we go. And we're going to look. I see some bubbles for sure. Oh, wow. Look at the one in the center. Look at the streaming just oozing out. Takes a lot to make a plant pathologist excited, right? Look, nope, this side. Look at the streaming coming up there. I don't have a good pointer. Let me see. I buried my mouse. Let me see if one of these bells and whistles will let me. Oh, let's see. I think I can. Nope, that's not working. Okay, so you can see my mouse? Oh, awesome. Okay, this is streaming, guys. Look, it's coming all around. It's coming down. This is all streaming. Look, it coming in this edge right here. You see how it's getting cloudy? Look here, more streaming. Get a little bit coming in here. You'll see all the dots all around this slide. That's probably dirty water that I'm using, scratched up slide. My slide's not clean. These are bubbles. So I didn't, see all these are bubbles, so I didn't get my cover slip on well. And these little lines here, that is plant tissue because my blade wasn't sharp enough. So what you're looking for is this cloudiness. Here's all this cloudiness. All right, that's all streaming. Here it is again, and we can move our slide around. Probably see it at different angles and different pieces. Let's look at that big piece. That bubble's really affecting it. But you'll see it around most of the edges. You'll see really great streaming. One thing we will look for, so some of these, so we're taking multiple pieces from multiple spots. You can't look at one piece and see what you're going to see. Sometimes we get lucky, but we don't always. So you're always going to use more than one leaf, more than one spot. You're going to make multiple copies, multiple pieces. And this is simply with a dyno light, okay? This is nothing fancy. This is not a professional microscope. This is not my lab scope. This is a dyno light. You can do these things. This backlight, remember we talked about in webinar 2, this backlight costs like $500 or $450, and it's just a little block with light. It's really convenient. It's a great size. I like it, but try it with your cell phone. And when we do our practicum in September, we'll bring this and we'll practice with cell phones too. So that way you can compare and contrast. So while I'm talking, I'll just kind of leave this here and look at that streaming. It's continuing. Those bacterial cells are building up right outside of that sample. And it's doing it for all of the 3 samples. Now, of course, I looked at this yesterday, so I knew that this was a bacterial pathogen. Had it not been a bacterial pathogen, you would not have gotten streaming. But it's building up, so if you look at it over time, it builds up. Any other questions there? Kim, do you see no chat questions? All right, so again, you can always use the button on your dyno light, and you can photograph these things. You can either share the screen with me, or if this is a vegetable you're going to share with Emily Pfeiffer, you can share the screen, you can snapshot these, and send the picture and say, is this bacterial streaming?
Do all bacteria stream?
Do all bacteria stream? Active bacteria stream. So for instance, I was going to show you fire blight in apple. It's sensitive to heat. It stopped streaming right now. It's too hot. We could have gotten it to stream earlier in the season, but we cannot get it to stream now. So active bacteria will stream. So if you're looking at anything in ornamentals or vegetables, you should absolutely have streaming right now. So when the pathogen is active, or when the when the disease is active and spreading, this bacterium will probably stream. So this is what the diagnostic lab is going to do. Okay. All right. Can I move off of this? All right. I'm going to stop sharing my screen for a sec. All righty. So I'm going to move it up. Okay. I'm going to show you this little block. This is just the little light block there. I just had my scope on top of it. This is just a little light block. That's all it is. And it just plugged right into the wall. And again, it was expensive. This comes from the Dyno Light company, but we can work with otherwise with them. All right. So another way we can stream. So we'll talk fire blight next. So I want to try to show you this. This is a fire blight canker. You can see, I'll let it adjust. So you can see the canker there. So when you would sample that canker, you would sample in the margin of the canker. Let's see if my blade is sharp enough. Now this fire blight is not going to stream right now. In the springtime, it absolutely would. Okay, my blade's not sharp enough. Apologies. But you would sample right at that margin of no, we're not focusing, are we? We're going to sample right at that margin, a very thin piece of wood, back onto that slide, and we're going to see that streaming. Okay, we may, in September, if it's cool enough, I may be able to find some fire blight. When it cools off, it might become active again. So you can do that. So I want to show you one other, well, let's see. No, we're going to have to back up. We're going to have to go in order. How are we here? So some other types of bacterial pathogens or fire blight, which I've already kind of told you that. Most people know fire blight from the shepherd's croak. Okay, so this distortion, this shepherd's croak, usually start seeing fire blight in June, sometimes earlier. And this is very distinctive of a shoot blight phase of the fire blight pathogen, Erwinia. So it's coming in through the tip, so you can usually cross-section this very narrow tissue in very thin pieces, and it will stream under a in water under a slide as well. So this is the classic fire blight symptom, but the way fire blight starts, as I showed you earlier, it starts in a spur. So this is where the flower spur was. You see the flowers, and infection came down, and that's where we see that canker coming in. And this particular piece, there are 3 of them. So the bacterium will move. Maybe I'll switch to Dino-Lite. I'm going to switch to Dino-Lite again, everybody, and see if I can get a... Oh, let me go the other way. Okay, so this is so awkward. See you. Bear with me. There we go. So you'll see the spur, and you'll see where the pathogen came down, and you'll see the margin coming in here of that canker. It stopped. All right, and then as you turn, see the cracking. Let's get the right way. You'll see the cracking here. Okay, and I have a younger spur. If I can get it to you. I have a younger spur. Get you in focus. You'll see the pathogen came down and it's starting. You have a smaller canker. You'll see it sunken here. So this is just not normal tissue. If you question it, you can always take your blade and check it. And again, I have a very dull blade. So the tissue there is green. So when you get into the canker, see, no, so it has not spread here. This is still nice and white and green. When the canker spreads, let's see if I can do this again. Try that. When the canker spreads, it's going to be brown. It's going to be brown, okay? That's your canker margin. We talked a little bit about that with fungi. I'm going the wrong way. We talked a little bit about that with fungi. You'll see another canker margin here. Where is that margin? There it is. Oh, I went too deep. Don't go too deep. You've got to stay really shallow. You got to stay really shallow. There we go. So that canker was moving. It stopped moving, in particular for this pathogen. Every pathogen has its own optimal temperatures and conditions. So the only other thing I want to mention about that, let me see if I can go higher, is that these cankers will spread more. I'm going to show you now. That's not going to work. You'll see the flattened edge of this canker came in at the spur. Came in at that spur broke off. There we go. This is a fire blight canker. This is what is going to kill trees, not exactly that tip blight. Okay? So I'm going to switch back off of Dino-Lite. There we go. So this is what I was showing you earlier. This is that fire blight canker. So it's pretty severe. Look at the flattened edge. Look where it came in at that spur, and it has just spread. There's another one that's come in here, and it has moved as well. So this was Molly's Delicious. pretty severe fire blight on that tree. And in time, so the pathogen will kind of shut down in the summer and it'll become active again when it's cool and rainy. Look at this guy. This is probably a 2-year canker, maybe 3. So this has continued and it's continued to spread. And of course, look what happened at the end. Okay, so this is really severe. This whole branch is dead. There was actually another canker below it as well. So this is why we prune out fire blight. This is sanitation. And you can always, during the active part of the season, which is usually April, May, you can section out very thin sections and you can see streaming, very good streaming. One other canker I want to show you. There is a peach canker, a bacterial canker in peach. There it is. It is not fire blight, but there is a canker in this peach. I can switch out to my dino light again. And this one is pretty specific to peach and cherry. Share screen. I'm gonna get you in. Here we go. Now you know peach ooze. So anytime you see a canker in peach, it's gonna be pretty oozy. So it's pretty gross in there. And you'll see that point of infection. The little white part to the right is where I just cut off a branch to get it out of the way. So peach are oozy, but this is a bacterial canker, not fire blight. It's different. So as this canker girdles the stem, it's going to cut off the xylem and phloem tissue, in particular the xylem, which is what's needed for the tip to grow. So you're going to see collapse of that stem. Some people ask me about fire blight in peach. It's not, but a bacterial canker often. We do get fungal cankers. So looking for streaming versus looking for fungal structures is one that we'll try to do for our September training. Okay, questions on bacterial cankers or bacterial leaf spots? Okay, so we're going to cover now, we're going to talk a little bit about bacterial wilt. Stop sharing my screen. Okay, any other questions? Feel free to unmute yourself and interrupt, and I know sometimes there's a delay. Oh, thread blight. No, thread blight is not a bacterium. Thread blight is a fungus. Thread blight in red thread in turf or thread blight in apple? Myron, thread blight on apple is really cool. Thread blight in apple, wow. No, it is fungal, so that's a really fun one. So I have to bring that to practicum. We did a project on that, so it's really fun. Okay, so our third type of, oh, I didn't show you water soaking. Okay, so I've got some peppers. So these are old peppers. This is a bacterial spot in pepper. So there's a bacterial spot. These are in really bad shape. So I'm telling you right now, my apologies, but I want to show you water soaking. I saw it yesterday. Let's see if I can find it again today. I'm going to do this again with a dino light. And I can share some pictures because I have photographed some before the sample collapsed. So usually when you see a bacterial pathogen, you're going to see what we call water soaking. Water soaking is the is the bacterium in the leaf. So I'm switching back again to share. And again, everyone, feel free to, oh, I lost my mouse. Feel free to interrupt if you don't. Okay, I am driving my mouse now in a puddle of water. How not to maintain your computer. Uh-oh. Okay, technical difficulties. Do you see it? It's over here. Oh, here we go. Jackpot. Okay. All right. I am going to show you what we call water soaking. Here we go. All right. Look at the wetness. You see that greasy or wet, kind of that blackening in that leaf tissue, that is a very extreme case of water soaking. Water soaking is usually really indicative of a bacterial pathogen. Okay, this is an old sample, but usually this is the bacterial cells accumulating in the leaf. It's rarely going to be this severe. You're usually going to see it much more minor situations, and it will usually be water soaking around a particular spot. Try to see something lighter, but this is a very old sample, so it's not showing the classic symptoms. But water soaking, imagine when you press a leaf and you damage the cells, when the cell contents are kind of floating around in that leaf tissue. So if you want to practice just kind of squeezing leaves with your finger, that's what water soaking is. So water soaking is something to look for. All right, as we go through our symptoms, remember any leaf spots, you're looking for halos, water soaking, and spots that do not have tan halos. Apologies, yes, you are seeing tan halos here because this is an old sample and the centers are falling out. All right, usually you don't have that tan color. So water soaking and yellow halos. If you have a wilt, that's specific to usually cucurbits and or tomatoes. Those are two different pathogens. It's usually in that cucurbit family that you're going to see the wilting, and we're going to cover that right now. Cankers, we see bacterial cankers in apple, and we see bacterial cankers in peach, different pathogens, but we see those cankers there. Usually when we see bacterial leaf spots, I'm jumping all over the board, we see them in the younger tissue, not as much the older tissue, and it waits until the leaf spots wait until it's hot, and it waits until it's rainy. So last week would have been a good week. Irrigated greenhouse is another one, whereas your fire blight, that's more of a cool season. So again, optimal temperatures. Okay, question?
Does water soaking occur on sunflowers? Trying to figure out what's going on with mine at home.
Sunflowers, we should not have a bacterial issue on sunflowers, but you would see water soaking. Zinnias, very common in zinnias. So this is what a zinnia leaf spot would probably look like. So yeah, you should look for water soaking and yellow halos, and then you can stream it. If you question it, stream it. And of course, if you want to double check yourself, give it to the send it to the diagnostic lab. Now, let me say that if you have a commercial sample, it should come to the diagnostic lab immediately, as soon as possible. If you want to play with an additional sample in your office, I highly encourage that, as a matter of fact. But a commercial sample should really go to the diagnostic lab because that's where the pros are. So we're just showing you how to help growers. Okay, before we switch out, I want to show you one more thing. I want to show you these yellow halos I did when we first got started today. This is, there we go. Here's some yellow halos, but this is something that is not bacterial. I would suspect, first of all, yes, this absolutely looks bacterial. This is Septoria leaf spot, but I see yellow halos, and I see spots on some of the leaves. There's not the white centers, but it's starting to develop the white centers. So this would look very similar to what you would expect to be a bacterial leaf spot, except we looked for sporulation. We saw sporulation here, and but we had to check it. So this would be suspicious. All right. I see the edges, but this is another thing. As a tissue starts dying, if it was bacterial, you couldn't see the spots anymore. Okay, but here you can still see the spots even through necrosis. Necrosis, again, is death of tissue. And so that's another indication. And this was the bottom leaves, which is where Septoria comes in. This was not the top leaves. So as you're filling out your diagnostic forms, it's really important for us to know where those symptoms occur. All right, and if the sample would have been brought into you and the homeowner or the grower didn't know, it's really difficult. It's really difficult for you to work. You have to make them responsible for going back and getting that proper information. So we'll be going through diagnostic forms. Okay, so the last thing I want to cover is bacterial streaming through vascular tissue. And by vascular tissue, I mean this is the cucurbit wilt. Let's see. I'm going to stop sharing. for a sec. All right, so I want to show you what came into the diagnostic lab. I don't know what this is. I think it's a watermelon. Not real sure. There's a fruit in here. No, I don't know what this is. Some type of melon. So this was a dead plant. The entire plant is here. Everything from the roots. And this is how you would submit a cucurbit sample, the entire sample. And it's twisted up into a mangled mess. This is an older sample. If this sample would have, if I'd have got my hands on this sample last week, we could have really played and done a lot of streaming. But unfortunately, it's old and dry. And this one has bacterial wilt. We're going to look at that right now. So I'm going to put this under the desk. Yeah, we've got that big piece. I'll put this under the desk so that we can address a few other things. Oh, I'm going to keep this guy. All right, get this out of the way. All right, so this is a disease that's pretty specific to cucurbits, and it is transmitted by cucumber beetles and other beetles. All right, I'm going to try to come up for you. Beetles transmit, so in order to suspect, the first thing we look for is beetle damage, beetle feeding damage. I'm going to see if I can do this. I might. No. No. Okay, I'm going to have to switch to my dino light. Sorry about all the switching. Share screen. All right, try again. Beetle feeding damage. Here we go. So you see the beetles have kind of gnawed there. This is the root. There we go. There's the root. And we see lots of beetle feeding up and down here. See the tan? And if you were here, you could see they had literally gnawed in. So the beetles will start feeding early in the season. There we go. That's a really good view. Beetles will start early in the season. So the longer that the bacterium has been in the plant, the higher the titer, or the higher the concentration. So when we're going to sample for the bacterium in this vascular wilt, we are going to look for at the base of the plant. This is the base of the plant. There's the root. You see the gnawing. And we are going to sample as low as we can. And we're going to look for streaming. Now, this is a cool trick. I'm switching back. Should I switch back out? So, okay, well, let me give an example. So what we're going to do is we're going to float. I have little vials. See the little vials? It's a little vial of water. So we're going to stream in a vial of water here. All right. So that's what I'm about to do. I'm going to toggle you back and forth. So what I want to do is catch at least an inch long piece. I'm going to go a little bit longer than that. I want something close to the base and something close to where the beetle feeding occurred. I like to use a branch because that branch holds the piece suspended in the vial. Do we have an empty vial? No? Okay, and I'm going to put my sample, and see, it's kind of like hooked right there. All right, and this is what is going to be hard for you. I am switching to a different stand for my dino light so I can get it from the side. Bear with me. There we go. And there we go. And we are going to look for streaming. All right. So this will be I can point with my mouse if I don't run it through the water again. So we are looking for the bacterial ooze coming out. All right. And I have already, now let's see. I want to look for, okay, we have some more streaming here for you. No, that's today's, is this today's pictures? This is what bacterial streaming will look like. Oh, that was a good one. Sorry. There we go. There we go. Here we go. This is bacterial streaming. This is what it's looking like. You'll see the bacterial cells coming out, and then these strings. Let me see. Nope. Did that come up? Okay, so you'll see the string happening. The first thing that's going to go on is the string is going to occur, and then it'll get longer, then it'll break off. So you'll see that, and then eventually, nope. And eventually there'll be, see, sorry. Okay, so here is ours. You'll see the movement. You'll see it building up. I'm going to switch out with you. Sometimes it'll take a few minutes. All right, let me get another one down here. Look at the streaming going on. There we go. And you'll see it's moving. I'll let this go, what happens. You'll see it's moving, so that streaming is happening. My dyno light is upside down. That's why this is happening here, because I'm trying to look at it from underneath. But that is streaming. And this is simply these little vials in with some water. And I will pan out. Is this another stream? Is this another good stream? All right. I'm going to pan out on these so you can see the other way as well. Now this is a little no. This is a glass container. Here we go. And I know there's a glare. That might help. No. Lighting is really a challenge. So this is a glass container that was scratched, and if you were here looking at it, you couldn't tell it was scratched. But when you start working with these lights and these cameras, it really becomes more difficult. All right, let's try again. No, that doesn't. It's my hand. Okay. So the file. There we go. Oh, let's see. You can see that streaming happening there. So you see the cloudiness, it's very severe, and you'll see it coming out and it's moving. Okay, there's Kim's got the mouse. So see, there's that puddling, that cloudiness, and then you'll see those strings starting to come out. So yeah, see it's going. And if you were looking at it in person, it would not be this difficult. We're just really struggling with lighting today. All right, so you can see my fingers. And so this is bacterial streaming. This is bacterial wilt in cucurbids. All right, so I'm going to pan out for you so you can see what the heck I've been doing. All right. There we go. Maybe. Let's see my messy office too. There we go. So this is kind of a fat piece. And if I stood still, I'm holding it in my hand. I stood still. So this is a fat piece, and it's just dunked in a little vial of water. You can see my hand. It's very small. All right. And here is the one that we did, the first one we did, in this little tiny vial. And this is the base of that. Backing up again. This is the base of that melon plant that we worked with. Is this a different one?
That's the one that you started.
Oh, this is the one that I started. It's starting to stream. Let's see if I can catch it for you. I moved it around, so that's not a good thing. It breaks off some of the strings. There we go. See it streaming. So how long ago was that? 5 minutes. There, look at that string developing. And it's breaking off. And another one being produced. So it takes a little bit, And then the second ones I showed you were the ones Kim started about 30 minutes ago, maybe. So if you're eating lunch or something, you could get them started and walk. But I did video one. I should have hit video for this, which will have the whole session recorded. You could go to it. So this is...
We have a question.
Oh, we have a question? Okay.
Is it best to cut the branch flat or at an angle?
Cut the branch flat and use a sharp blade. Don't use your pruners because that just crushes the tissue. So use a very sharp blade. And you know, you buy razor blades by the what, 100 in a little box. So don't hesitate to throw blades away and start again. So if you work with vegetables in particular, this is a great technique for you. If not, it's just fun. All right. Everybody loves to do this. I'm going to back up. I'm still holding. Here we go. So these are fairly small pieces. I'll put my ink pen here so you can just see perspective. So they're not big. Okay, so we have small vials. It's just water. And yeah, so half the length of an ink pen. Very small vials. You could use anything. I highly recommend glass. Okay, I'm going to turn this off and stop sharing. I highly recommend glass. Glass is clear, and you can definitely see. What's the remote to this thing? There we go. There's our operation we've got going on here. Little vials, little tiny. This was a bigger one. This was the base of that root. So it has a little wider mouth. And we're gonna put and here's the rest of that plant. This is the root. This is where I cut it off. And this is where the sample came from. Okay? Questions? All right, we're getting to the end of our time. We have 5 minutes left on our time. So I'm happy to answer any questions, review anything you want to review. It's your choice. Get up so you can see my face. So I was using this this stand just so I could bend underneath. This is the stand that came with all of your dino lights. This is probably the first time I've ever really used it and found benefit in it. And then this was, remember, the nicer stand that we purchased, $140 if I remember right. $140. Oh, you want to do that? Is it? No.
It's kind of doing it.
Okay. All right. Backtrack. So this is another technique that you can use in a fresh sample for your to look at streaming, is when you cut a piece. So if you've got a bacterial population that's accumulated, I know you're not going to be able to see this. If you touch it together and you pull it apart, you'll see stringiness that connects. And I could send you some literature on that. So this would be very classic when that sample fresh came to you. If you want to do this, remember, diagnostic lab needs a fresh sample, the entire plant, root and everything. So you're not going to cut this and play with it and then send it to the diagnostic lab. So ask your grower for a couple of plants for the diagnostic lab and a couple of plants for you to experiment with. So you're going to get this stringiness happening here. And this came also from the base, and we'll see some beetle feeding here. So we know there's an accumulation of enough bacteria to quickly diagnose. Okay, so let's review some things we learned today. We learned about spots, bacterial spots. They're angular. They're typically in the newer tissue. They come in warmer weather. They rarely have a tan center. They will appear usually where a drop flows, in clusters, or in the edge of a leaf. You will often see water soaking, and you should be able to get streaming if it's active. All right, our second group of bacterial diseases were the cankers. Typically, we know peach has a bacterial canker and apple has a bacterial canker. The apple is fire blight, the spur blight phase, and the peach, we call it bacterial canker. They're different pathogens, but they both make cankers. You can also stream thin pieces at that canker margin. And then the third group of bacterial symptoms is the bacterial wilt. Here in Kentucky, we're particularly talking about our cucurbit group. And that's where we can stream near the beetle damage at the base of the plant. And we'll see collapsing pretty quickly. So that's when you would take that sample. Be careful. Bacteria is kind of hard. There are some fungal pathogens that look like bacterial spot. There are some fungal cankers that you can easily mistake for. That's why we look for streaming. And so don't make an immediate diagnosis that just from symptoms. So you're going to have to look for streaming. If it's a commercial sample, you're always going to send a sample to the diagnostic lab. You could play along at your desk. You could, and if it's vegetables, you're going to stream in with Emily Pfeiffer. If it's ornamentals or fruit, you could stream in with me. I'm very happy to do that, and we can help you confirm that way, especially if it's a homeowner sample. Any other questions? We've got a minute, or I can stay on longer if anybody has any questions. Link for the quiz. Link for the quiz. All right. I am sharing screen again. Link, where did it go? Here we go. Why is it doing that?
It's showing it.
It's showing it? Oh, it's on my other screen. All right, hang tight, guys. Yeah, there we go. There is our quiz. Oh, remember, plantdiseasetraining.ca.uky.edu is where you're going to find this webinar and others. And there is the link for the quiz. Kim is going to put it in the chat box.
And we have a question, which is,
Kim, okay.
Would sap ever look like streaming?
Would sap ever look like streaming? No, sap will not look like streaming. Streaming is white and it'll build up because it's much thicker. Sap is water-based and it's going to just dissipate and you really won't see it. So sap is not going to be confused with streaming. And if you wonder, find a healthy plant of that same species and put it in.
What about gummy stem?
Gummy stem, I don't know. I don't know that one. I'm not the veggie pathologist. I will get back to you on that. So that's a question that I will follow up with. Sorry about that. All right. Other questions? I can't see the chat when I'm sharing this way. Let's see. There's no other way to. Can I show taskbar? No. Okay. Any other questions? Okay, again, you have to get the quiz in. You have 1 week to get the quiz in. It's simple. It's simple questions. It's just our way of verifying and of documenting. There are 5 questions. I think there are 3 bacterial questions, and then there's the comment question. So we always want your comments to come in. And again, apologies that I didn't follow up after the last training. I will get that to you. I will get vocabulary in as well. We'll start building on. Let's see, can I go this way? There we go. I will start building, and if you go into the webinar, if you click webinar, there are some resources. I'll start building onto that resource. Next webinar is July 20th, and it's going to be oomycetes, which are your water molds. It's Pythium, Phytophthora, downy mildews. That's where those are going to appear. So plantdiseasetraining.ca.uky.edu. It's in all of our emails that pertain to this program. And there is your quiz. And if you have any problems, the sooner I know, the better. We can fix it and we won't have any problems. All right. I am going to stop recording now, everyone. Let's see. How do I do that? Okay, I have to come off. And I'm going to